everyone, and welcome to this entire topic video for topic 3 GCSE Biology. This is going to be a long one, so don't forget you can watch it at ×2 speed to get through it a bit quicker. And if you do want even more help boosting your grade, check out my GCSE notes linked below. They are checked by examiners, they're mark scheme specific, they cover all of the theory, and they've also got key terms, summaries, questions and answers as well. So best of luck with your exams everyone, and don't forget I've got a video like this for every single topic for GCSE Biology. Best of luck. So topic 3 is all about infection and response, and we start with looking at communicable diseases, which is an infectious disease, and looking at the different pathogens that you need to know. Pathogens are microorganisms, so they're microscopic living things, but the difference between a microbe and a pathogen is it is a microbe that could cause an infectious disease, and that is in animals and in plants. These diseases can spread between organisms, which is why they are called communicable diseases. Pathogens need a host, which is an organism they infect to survive, and the host provides the right conditions and the nutrients, such as glucose, for the pathogen to grow and reproduce. There's two main ways that they can make you ill. Number one is producing toxins. Bacteria release poisons, which is what a toxin is, and these toxins can damage your cells and your tissues, causing the symptoms that you might get, such as a fever, pain, or even swellings. The other way that they can cause harm is through attacking your cells. And viruses do this by invading your body cells to reproduce, and then as they reproduce, they damage and destroy your cells, and that can also lead to symptoms and illness. The body does have natural defences, such as your skin and your immune system, to protect against these pathogens, which is what we're going to be going through later in this topic. But first, let's have a look at the pathogens in a bit more detail. So we already said what a communicable disease is, and they're caused by different pathogens. The pathogens that you need to know are viruses, which you can see here in the top right. These reproduce quickly inside of cells, and that is how they cause damage. Bacteria also reproduce quickly. About once every 20 minutes they reproduce, and they can release toxins, and because you have so many bacteria producing those toxins, it can make you feel ill. Protists, which we can see here, these are a group of microorganisms that can cause disease. Some are parasites, meaning they live inside of a host and cause harm, and that is malaria as an example, which comes up later in this topic. And then lastly are fungi, and they can grow on the surface of organisms, and they sometimes release toxins as well. So the way that these diseases are transmitted, or in other words, how the pathogens can be spread between different organisms, varies depending on the type of pathogen it is and the disease that they cause. The three modes of transmission that you need to know about are direct contact, transmitted through water, or transmitted through air or droplet infection. And this table summarises what these types of transmission mean. So for direct contact, this means that it is spread, the pathogen is spread through physical contact, and that could be between touching an infected person, an infected surface or object, and this can be the case for plants as well. If you have an infected leaf falling onto a healthy plant, we've got an example here of athlete's foot, so that spreads through sharing towels that then will have the fungi on them, or walking barefoot in damp places where you might have the fungus there already. So to prevent getting infections through direct contact, you should avoid sharing personal items and wash hands regularly. Water is when the pathogen is carried in contaminated water. So that would enter your body through drinking, or it could be through bathing, and then if you had a cut in your skin, it might be able to enter that way. Cholera is an example, and that is spread through drinking unclean water. So to prevent this, there are strategies such as boiling water before drinking it, or using purification methods, and ensuring good hygiene and sanitation. Lastly, we've got the air droplets. This is when the pathogen spreads through air in tiny droplets when an infected person coughs or sneezes. And this is how the common cold, flu, COVID-19 are spread. It's through inhaling those virus-containing droplets. So to prevent the spread, cover your mouth when you are coughing, when you're sneezing. Wearing masks prevents the spread as well. And in crowded areas, try and make sure that you are having as much ventilation as possible. So here we have a little bit more detail on preventative measures. We did mention some of those on the last slide, but to have a look at some of them in more detail. Good hygiene, so we've got washing hands, but In an exam, you need to be specific. Washing hands with soap, because just with water isn't going to remove the pathogen. You have to be using a soap. For vaccines, that is going to prevent the spread of infection as well, because if you have fewer people able to have that infection, then there's fewer people to pass it on. Using disinfectant, so this is a chemical that kills bacteria and you use it on surfaces. So you could be cleaning surfaces regularly with disinfectants to kill bacteria. It can also destroy some viruses, and therefore if you're touching that surface, you aren't going to be touching anything with pathogens on. Food safety is another one, because if you aren't cooking food through properly, particularly meat, then pathogens such as salmonella can be in that food, and then when you eat it, the pathogen gets into your body. The final example we've got is quarantining people who are infected. So that means people that are infected, they'd be kept separate from healthy people so they can't transmit that pathogen. And that's something that we saw a lot during COVID-19. So that's how we can reduce the transmission of the diseases. You also need to know about the pathogens in a bit more detail and some different examples. So starting with viruses then, they will invade and reproduce within a host cell. And when we say host cell, that means the individual that they are infecting. And they have to be inside of a host to be able to replicate. And because the virus reproduces inside of body cells, that then causes the body cells to burst and it causes damage. It also means it's quite difficult to treat the virus, because in order to treat the virus, you would have to destroy your own body cell. Antibiotics, which are a type of medicine, do not work against viruses. So treatments to try and remove a virus from your body actually focus on preventing infection in the first place, because it's very hard to treat a virus once you're infected. You have to then just rely on your own immune system to destroy the virus. So things like vaccines prevent infection, and for certain viruses there are antiviral drugs as well, which will help. The three examples that you need to know are measles and HIV, which both affect humans, and then tobacco mosaic virus, which affects plants. Next, if we go on to bacteria, bacterial diseases are caused by bacteria, that pathogen, which are single-celled microorganisms. And unlike viruses, bacteria can live and reproduce independently in the right conditions. So that means they don't have to be inside of the host to reproduce, and for that reason, bacteria can actually survive on surfaces. Some bacteria are harmless and some are actually beneficial to humans and other organisms, but if it does cause harm, it's known as a pathogen, and you do get pathogens that are bacteria that infect both animals and plants. The two key ways that they cause harm are by their rapid reproduction, increasing the number inside of you, and then you've got so many of them producing toxins, which are poisons. And those toxins could damage your tissues, or they could cause symptoms like we talked about earlier, like fever, vomiting, and even diarrhea. The examples that you need to know are two that infect humans, so salmonella and gonorrhea. The next group that we're going to look at are fungus. And fungal diseases are caused by fungi, which can be a type of pathogen. And that could be mold, yeast, mushrooms, and you have some that attack animals and also plants. Fungal infections are often spread by spores, which are a bit like tiny little seeds in fungi. And they can travel in the air, water, and also they can remain on surfaces. And the key one that you need to know is a fungal disease that infects plants known as rose black spot. Lastly then, we've got the protists, and these are single-celled microorganisms. They are not bacteria, they're not viruses or fungi. Some of them are parasites, which means they have to be inside of a host to survive and then cause harm. The one that you need to know about is malaria. Now, as we went through all of those, I said the ones that you need to know about, and I said the names of them and whether they infect animals or plants. But this slide now gives you the table of how they're transmitted, the symptoms that they can cause, and the prevention and treatment. So I recommend you pause, screenshot, and then you've got all of this information for your notes. These are just key facts that you're going to need to remember for the exam. So you've got the disease, the pathogen that causes them, how they're transmitted, symptoms, and also how you can prevent and treat them. You might want to also add whether it is a plant or an animal that has been infected, and that was on those previous slides if you want to add that in as well. So next then we're going to have a look at how humans are able to defend themselves against pathogens. And there's two key types of defences. We have something called the non-specific defence system. This is like your first line of defence. That will be the barrier and protect against any potential pathogens. And that could be preventing them from getting in, but also how they're destroyed if they do get in. And it's called non-specific because it's going to be the same strategy, the defence system, no matter what the pathogen is. Next then we've got the immune system, and this is a targeted defence that fights specific pathogens if they do manage to get into the body. So let's have a look at some of these non-specific defence systems and also the barriers you have to entry. So non-specific defences are there to block or remove pathogens before they can cause any harm to your cells and tissues. These defences are always active and you do not need to recognise the particular pathogen in order for them to work. So we can see here we've got the skin. The skin is a physical barrier that stops the pathogens from entering. The skin also has sebaceous glands within it, which produces oils, and some of those oils are able to kill bacteria. If you get a cut in your skin, then you will quickly form a blood clot and then a scab, and that is to prevent your barrier, the skin physical barrier, from having any holes in, which could then enable a pathogen to enter. Next then we've got the nose. This contains hairs and mucus that can trap dust, but also bacteria and viruses to prevent them from entering your lungs and causing damage. You also have that reflex of sneezing, or you might blow your nose to remove any mucus in your nose that would have potentially trapped pathogens within it. Next then we have the trachea and bronchi, which are part of this breathing system. They are lined with mucus and also cilia, which are hair-like structures. The mucus is a thick, sticky substance which will trap any dust, but also bacteria and viruses. And the cilia are these tiny hair-like structures that move in this sweeping motion. So it's a bit like they are a broom sweeping the mucus up and out of the trachea, and you then end up coughing it, or you might swallow it into the stomach. Either way, it's going to prevent it from getting to the lungs and therefore causing any damage to the lungs. Lastly, the stomach. The stomach has hydrochloric acid within it, which is a very strong acid. So we just said that the cilia will sweep the mucus up and you might cough it out or you might swallow it. If you swallow it and it's going into your stomach then has hydrochloric acid within it, which can kill most pathogens. So it's going to help to destroy any pathogens in swallowed mucus or in any food or drink that you have eaten. Now, if a pathogen does get past those responses, we then move from what was non-specific to specific defense against pathogens. And this is where your white blood cells come in. So your white blood cells' roles are to help to destroy pathogens and therefore prevent damage to your cells and tissues. And there's three key things that white blood cells do. Number one is phagocytosis, and this is when a white blood cell can move around or engulf a pathogen. And once it's engulfed the pathogen, it releases enzymes to destroy that pathogen, such as a bacterium. And then it's destroyed and it can't cause harm. White blood cells can also produce antibodies, and antibodies are these structures that are complementary in shape to antigens, which are structures on the outside of pathogens. So your white blood cells produce antibodies. The pathogens have antigens on the outside, and when the antibodies bind to those antigens, it makes it easier for these white blood cells, the phagocytes, to locate and destroy the pathogen. Lastly, there's antitoxins, and we talked about how bacteria produce toxins, these poisons, which cause harm. So white blood cells can also produce antitoxins, which are complementary in shape to the toxin, which means it binds to them. It therefore stops the toxin from binding to your body cells, and that means the poison, that toxin, can't cause harm. So here we have that information I was just saying now written out. So if you want to pause, take a screenshot, you can then add that into your notes or turn it into flashcards. Those extra details going through what phagocytosis, antibody production, antitoxin production are, and how they help to destroy pathogens. So another way to help prevent the spread that we talked about was having a vaccine. So vaccines are a preventative measurement and they protect individuals from contracting a particular disease. It involves introducing small amounts of either dead or inactive versions of the pathogen that causes the infection, and that could be through an injection. These pathogens that are injected in can't actually cause the disease themselves, but they can trigger an immune response. So let's go through how a vaccine works, because this is quite a common long answer question. So mark number 1 would be for saying that in a vaccine, a dead or inactive form of the pathogen is injected into the body. The immune system will then detect that injected pathogen and the white blood cells respond. The response of the white blood cells is to produce specific antibodies, and the mark scheme is really particular that you do have to say specific antibodies because it's all to do with complementary shapes fitting together. The body then remembers that particular pathogen and therefore the specific antibody to create. So if you are reinfected with the same pathogen later on after your vaccine, the immune system is able to respond really quickly and create large quantities very rapidly of that specific antibody. That means that those antibodies are going to bind to the antigens on the pathogens, and that means that the pathogen can't cause harm and it will be destroyed before it can cause any illness, or in other words, it's destroyed before you get any symptoms. Or you might just get very, very mild symptoms rather than getting the full-blown symptoms. So a bit more then on how that works. After you've been vaccinated, what happens is, as we said, if you are reinfected, you get large quantities of the antibodies being produced very, very rapidly if you're reinfected. And that's what this graph is showing us. At this point where it says initial exposure, what that would mean is that is when you were injected with the vaccine. So that was the first time you were exposed to that pathogen. It was just a weakened version of it. And your body's responses over time, you've produced a small amount of antibodies. And that will be enough to just remove the small amounts of inactive or dead pathogen that was injected. But then your white blood cells have a memory of that particular pathogen and antibody to produce. So if later on you do get infected with the actual pathogen, that would be our second exposure, because your white blood cells already have a memory of that pathogen and the antibody, it's able to much, much quicker, and that's why we've got a much steeper curve here, much quicker and in larger quantities to produce those antibodies so that the pathogen will be destroyed before you get any symptoms. Now, what that means is you have this long-term immunity to that particular pathogen. And you having these vaccines, or a population as a whole, not only does it protect you from getting that disease, it helps to protect the whole population because of something called herd immunity. Now, you don't need to know literally that term herd immunity, but you do need to know that if enough of the population are vaccinated against a particular disease, it will reduce the spread of that pathogen. And that is because if a large enough amount of people are vaccinated, that means all of the unvaccinated people, which would be very few, they're the only ones that can now get infected and pass on the disease. So if most people can't get the disease and get infected, they can't spread it, that should therefore protect the people in the population who are not able to have vaccines. And that is sometimes pregnant women or very early newborn babies or anyone with a weakened immune system might not be able to have a vaccine. So if a large enough proportion do have the vaccine, it protects those vulnerable people. And that's why there was such a drive during COVID to try and encourage people that are healthy enough to have the vaccine, to try and protect themselves, but also to protect the entire population. Now, another treatment is antibiotics. Now, these are medicines that only kill bacteria. They do not work against viruses, and that is because viruses reproduce inside of cells, but also viruses aren't actually living things. So the way that antibiotics work don't work on viruses. So the way that antibiotics work on bacteria are that they can either break down the cell wall of the bacteria, and that causes them to be destroyed. And also, some antibiotics will stop the bacteria from growing and reproducing. So different antibiotics are used for different bacterial infections, and they do work in different ways. Now, before antibiotics, bacterial infections like pneumonia and tuberculosis were often fatal. And since the 1940s, when antibiotics were discovered, antibiotics have saved millions of lives. But there has been a downside to this because the misuse and overuse of antibiotics has resulted in some bacteria being antibiotic resistance. So let's go through what that means. Some bacteria have mutated, and what that means is their DNA, so their genetic material, has changed. That's what a mutation is. It's a change in the DNA. And that change happened to give the bacteria a slightly different either cell wall structure or way that they grow and reproduce. And as a result, the bacteria that had that mutated DNA were now not affected by the antibiotics, or in other words, they are resistant to them. So the antibiotics will not kill that bacteria. Now, these mutations randomly happen, so it's completely spontaneous or random, it's due to chance, but the overuse or misuse of antibiotics made this process happen more quickly. And the reason for that is, if there was a bacteria that had this mutated DNA, which meant it could now survive if antibiotics were taken, if there was someone who was taking lots and lots of antibiotics, even though maybe they didn't need them, for example, when they were infected with a virus, that means that antibiotics would be killing all of the non-resistant bacteria, and just the resistant bacteria are left in your body. And now they don't have to compete for the resources. They can reproduce and thrive. And suddenly you now have a large population of resistant bacteria. Now, you don't actually need to know this, but an example is MRSA, which you may have heard of. So MRSA, this long name you definitely don't need to know, but methicillin-resistant Staphylococcus aureus, that's just one example of a bacteria that is now resistant to many different antibiotics. So how can we try and reduce this? First of all, only use antibiotics when necessary. And by that we mean it has to be a bacterial infection, and you should only use it if your own immune system isn't able to destroy the pathogen itself. You should also always complete the full course of antibiotics. So if you're given antibiotics to take twice a day for 7 days, but you felt better by day 4 or 5, you should still take all of those antibiotics to kill all of the bacteria. And also, doctors now avoid prescribing antibiotics, and if it's a viral infection, they won't. New antibiotics also need to be developed to help to replace the ones that no longer work because bacteria are resistant to them. There's another type of medicine that you need to know about, which are painkillers. So paracetamol, ibuprofen, aspirin, you might be familiar with. And these do not kill the pathogen. So they're not going to cure any illnesses, but what they do is relieve symptoms, as the name suggests. They kill pain. So not only can they reduce pain, but some painkillers also bring down a temperature. So if you've got a fever, some of them also reduce swelling, so inflammation in the body. Unlike antibiotics, though, as we said, painkillers do not treat the cause of the disease. So let's have a look at how some of these drugs were discovered, and then we'll go on to development. So for our drug discovery, most of our early medicines were coming from plants and microorganisms. And that is a particular chemical that you might find in a plant or a microbe. And there's 3 examples that you need to know. Digitalis, and that comes from this flower here, foxglove. That is a chemical that is now used as a heart drug. Aspirin, that is a painkiller originally found in willow bark. And then penicillin, which is an antibiotic. It was actually the first discovered antibiotic by Alexander Fleming. And that was in this mold, Penicillium mold. Modern drug development, though, is actually more so focused on synthesizing, which means creating new drugs by chemists in these huge pharmaceutical companies. So it's less so about discovering something by chance in plants or in fungi. It's all about using your chemistry knowledge to design a particular drug that might treat the disease. However, many drugs do still have the same start point of knowing a particular naturally occurring chemical in plants or microbes, and then scientists can edit and modify the chemical to try and make it more effective. So how are new drugs tested then? Because once a drug has been discovered, it can't actually be given to people straight away. And it often takes around 12 years between the discovery of a drug before that drug is commonly prescribed. And that's because there has to be several tests that are done to look at, first of all, toxicity. That means testing, is the drug toxic? Is it going to be harmful and cause side effects? Efficacy, that means does the drug actually work? So is it going to help to cure or prevent the disease? And then lastly, the dosage. You have to make sure that you know exactly how much different people should be taking so that the drug is effective, but also so it is safe and not causing harmful side effects. So there's two main stages to these drug testings. Preclinical trials comes first, and that means trials that you do before you test it on any humans. So step one is just testing it in a lab, and you'd be testing this new drug on just groups of cells or tissues and then live animals, so things like mice, for example. And the point of these stages are to help to look for any potential side effects. So it's testing for toxicity and looking at how the drug interacts with body cells. If the drug passes this stage, meaning it didn't cause any harmful effects to the cells or the live animals, then you go on to step two, which is clinical trials, which is now when we test the drug on humans. So once it's past the animal and cell stage, which is preclinical testing, then you test on humans. So this involves testing healthy volunteers and also small groups of patients as well to test, is it safe on humans, and also is it effective? So these clinical trials on humans, so testing on humans, are split into different phases. Phase one, you give a very low dose of the drug to healthy volunteers. And we do it as a low dose because if there are harmful side effects, at least you've only given them a low quantity, so hopefully it doesn't cause too much harm. And it always starts on healthy volunteers. Phase two, the drug is then tested on a small group of patients. And again, they will be volunteers, but they are patients. And this is to see if the drug does actually treat the disease. And we only test a small number of people just to make sure it's not going to be potentially harmful. And if it is, it's only affected a few people rather than lots of people. And then lastly, phase three, this is where we move on to larger trials on many patients. This is going to help to determine the optimum dose. There's also this concept of double-blind trials, or you might have heard of a placebo. And this is when you do all of those drug trial stages, the clinical trials on humans. Some patients receive the actual drug that you are testing, while others get a placebo. And a placebo is a fake or a dummy treatment. So you would give them what looks like the medicine, except it doesn't have the treatment in at all. So for example, if you were giving someone a tablet, which was the new drug, you would give someone instead a tablet that looks identical, but it would just be a sugar pill. So that's all it contains, sugar. And in these double blinds, neither the patients nor the doctors know who has received the real drug. And that's why it's called double blind, because the patients and the doctors are blind. They don't know who has had the real drug versus the placebo. Now, you do have this information written down. It's just you find that information out afterwards, or you have an independent person who will have that information. And the whole point of doing this is to avoid bias, because if you are told you now have this new drug, sometimes the power of the mind is enough to make you start to get better or feel better rather than the drug itself. And with the doctors knowing, without realizing, they might ask leading questions. So they might say to the group with the drug, are you feeling better now? Whereas they might say to the group that have had the placebo, have you noticed any change? And just those slight differences repeatedly in the way you might phrase it is enough to potentially sway how someone feels. Now, as well as these double blinds to try and make sure that we've got really accurate information, there's also something called peer review. And peer reviewing is once you've done all of these clinical trials and you've got all your information and you might come to a conclusion as to whether the drug is effective or not, you also have to have other scientists check the work to make sure that they agree the method that was used is valid and that they agree that the scientists have analysed the data correctly. So results of drug testing and trials are published only after peer review, and this is to make sure we have accurate and reliable data before new drugs are approved. So peer review also helps to prevent any false claims or even errors that people might be trying to put out there from their research. This next section on monoclonal antibodies is only on the higher tier. So if you're not doing higher tier, this section is not going to be relevant for you and you can skip to plant disease. So what are monoclonal antibodies? These are identical antibodies, that's the clonal part, they're identical, and they target one specific protein. And that's what the mono is, one. So identical antibodies that target one specific protein antigen. They're used to identify or treat specific cells in the body. So how they are made is another common long answer question. It could be 4 marks, it could be more. I've split it up here as if it was a 4-mark question. So let's go through it. First of all, why do we want to make them? Well, we make them in large quantities and we're going to look at the uses on the next slide, but we want to create a large quantity of these monoclonal antibodies. So step 1 is stimulating the mouse lymphocytes, because the mouse is going to be like our production center. So a mouse is going to be injected with the antigen that you want to make antibodies against. So it's going to be injected into the mouse. The mouse's white blood cells, specifically a lymphocyte, will then produce antibodies against that antigen, meaning complementary in shape. Next step is we need to create what's known as a hybridoma cell. Hybrid meaning 2 different things fused together. And what we're fusing together is this lymphocyte we've just created that creates the correct shaped antibody and a tumour cell. So those 2 are fused together to make a hybridoma. The reason for that is this lymphocyte is going to create the specific antibody and the tumour cell rapidly divides. So that now means we've created a hybridoma cell which will rapidly divide. So we create large numbers of identical lymphocytes that produce the specific antibody, that same specific antibody that we want. And we then allow enough time for it to produce lots of those antibodies, collect them, purify them, and we now have a large quantity of monoclonal antibodies. So the uses of those monoclonal antibodies, they could be used in pregnancy tests. You don't actually need to know the details of how it would work in a pregnancy test. It might come up as an application question, but the idea is you are creating monoclonal antibodies complementary in shape to a hormone that you only make when you're pregnant, and it's a way to identify if that hormone is in your blood. You can also use them to measure substances in the blood. So sometimes it might be for drug testing. It could be to identify molecules in cells or tissues, and sometimes it can be used to treat diseases as well, because you could make these monoclonal antibodies complementary in shape to cancer, so antigens on the outside of cancer cells, and that would then mean that the antibodies are only attaching to the cancer cells, and you can then attach a drug to the antibody, so the treatment goes directly to that cancer cell. So here we have it in a little bit more detail. Again, like I've said previously, you can pause, screenshot, and then you've got that extra information here if you want to add it to your notes. Lastly, then you need to know about the advantages and disadvantages of using monoclonal antibodies. The advantages are they are really specific, which means they will only bind to one type of antigen, and therefore you can use them as really targeted ways to identify molecules or to send treatment. And that's what this next one is. It can help to target cancer cells So you'd send the medicine directly to the cancer cell through this monoclonal antibody, and therefore it will destroy the cancer cell, but it won't harm as many healthy cells. And as we said, it's useful because you can use it in pregnancy tests to help diagnose if you are pregnant, or diagnose isn't the right term for that, but to detect if you're pregnant, but also diagnose for diseases as well. Disadvantages: it is expensive to produce and it does require advanced technology. They've actually found that using this as a treatment for things like cancer has had more side effects than they were expecting, particularly allergic reactions. And we've also got, it's not widely used yet in terms of treatments. It is in terms of testing for disease and pregnancy though. The final part of topic 3 is on both the higher and foundation paper, but is not on combined science. This is for biology only, meaning the separate sciences or triple science. So detection and identification is one part of this topic that you need to know. So how can you actually tell if a plant is infected? And plants can also show visible symptoms when they are infected by pathogens like viruses, bacteria or fungi, or even insects, which isn't a pathogen, but they can still cause harm. So here are your common side effects or symptoms. You've got the visual here and then also the list. So stunted growth just means it's not growing properly. Sometimes you see spots on the leaves, so discoloration and in patches. You might have areas of decay where parts of the leaf or the plant are starting to die and break down. You might have growths, which mean these abnormal lumps, which could be on the leaves or it could be on the actual stems as well. You might get malformed stems or leaves, so they have a different appearance. It might be a bit twisted. Discoloration, so they might be yellow instead of the usual green. And then the presence of pests. So if you can see lots of aphids, for example, that is an indication that it is infected with aphids. Now, these are the common plant diseases, the symptoms and the causes that you need to know. So you might want to screenshot, turn this into flashcards, or just have this table in your notes. But tobacco mosaic virus we had come up earlier on. That is a virus. It causes these mosaic patterns on leaves. It stunts the growth. Rose black spot is a fungal infection. It causes these purple black spots on the leaves. And in both of these cases, because you are damaging the leaf, that means you have less photosynthesis occurring, less glucose being produced, less glucose for respiration. That means less energy, and the plant just isn't going to grow as much. Aphid infestation. So aphids are an insect. They're a type of pest. This also results in stunted growth. You might even get wilting, which is when the leaves start to droop, and you might get yellow leaves. And the cause is the aphids are biting into the plants and they are feeding on the plant's sap, which is the sugary substances. And if they're using up all of those sugars, then that means the plant doesn't have the sugars like glucose it needs for respiration. Then we've got the two at the bottom, which aren't infectious diseases. These are deficiencies, meaning you don't have enough of something. And nitrate deficiency is when a plant doesn't have enough nitrate ions, which are an example of minerals that they get from the soil. The nitrate ions are needed to make proteins. So if you don't have enough nitrate ions in the plant, it doesn't have enough proteins, and that means it's not going to grow as much. So you get this stunted growth. It looks smaller. Magnesium ions, again, it's absorbed through the root hair cells from the soil. And magnesium ions are needed to make chlorophyll, which is the green pigment in leaves. This is why the symptom of magnesium deficiency is this yellow coloured leaf. And because it's not got chlorophyll, it can't absorb light energy, it's not going to photosynthesize as much, and therefore it's not going to be producing the glucose it needs for respiration. And again, you will end up then getting a smaller plant eventually. So how can plants defend themselves then? Because they don't have an immune system, they don't have white blood cells, but they are still able to defend themselves. So you have physical defenses, and those are, for example, the cellulose cell wall. That helps to protect the contents of the cell. Leaves have a tough, waxy cuticle to prevent entry into the leaf. And also bark does start to peel off. And as it peels off, that means that any potential pathogens that might be on the bark are going to fall off. You then have chemical defenses, and some plants, such as witch hazel, which is what this picture here is showing you, Some plants produce chemicals which can kill pathogens. Lastly, we've got mechanical defences, so things here like thorns. Now, thorns do not defend against pathogens because pathogens are microscopic, so a thorn is not going to do anything to them. But what these thorns do is prevent animals such as herbivores, so large animals that eat plants, from being able to eat the plant. So let's have a look at this summary here. Here's our type of defense. Here's some examples, and here's a description. All of these examples of defenses you need to know. So I've talked through some of them. So we've got our physical ones, which we saw on the previous slide with the picture, and you've got the description. We talked about having those chemicals and also poisons that can help too. First of all, the antibacterial chemicals will destroy pathogens, but also some plants produce poisons which are toxic to animals, and it prevents an animal from eating them. Mechanical, we talked about the thorns and sometimes hairs, but there are also other mechanical defences. Some leaves will droop or curl when touched, and when that happens, it can scare an animal off because they might think it's another animal as well. So it's to help to prevent herbivores eating them. And this is the same idea with mimicry. Mimicry is the fact that some plants actually look like dangerous animals or other plants to trick animals to avoid them from eating them. So that takes us to the end of topic 3. Pause now and have a go at these end of topic questions just to see how well you remember and have understood everything. And then on the next slide, we'll have a look at the answers. Any that you didn't remember or you got wrong, go back and watch that part of the video again to try and help consolidate that information. But you pause now. I'm going to move straight on to the answers. Here are the answers for those 10 questions. So pause, mark your answers and see how you did. If you have found this helpful, then my GCSE notes are starting to become available one topic at a time. And all of the notes include key marking points, examiner tips, so where I tell you little tips of key terms to include or mistakes to avoid to help secure you get top marks, as well as you get the topic summaries, you get end of topic questions and answers different to the ones that feature on this video, key terms as well, and the definitions within so that you can make your own flashcards. The link's in the description for those. But for now, that is it. I hope you found this video helpful, and I will see you in the next video, which will be topic 4.